Hello my dear learners, welcome to Learn Microbiology for another video on the series What Next After BSc Microbiology. In today's video, we will be solving the mock test of GAT-B part 3. Let's get started. Question number 14. Cell cycle controller is robust and adaptable. Additionally, it functions like a switch to ensure unidirectional cell cycle. This switch-like behavior can be achieved by which of the following mechanisms? As we can see here, the question is about the cell cycle. We have understood the cell cycle in the previous video. We know cell cycle consists of 4 phases. One is G1, S, G2 and M phase. Now, this cycle is controlled with the help of a regulation mechanisms which is robust and adaptable. That is, this mechanism can be adapted based on the environmental signals. Now, we also know that this particular cell cycle is unidirectional, that is, it can occur only in one particular direction, that is a clockwise or an anti-clockwise direction. Now, there is a switch to ensure this. Switch-like behavior is achieved by which of the following mechanisms? There is a protein complexes which is called as cyclin and CTK. Cyclin is a protein, CTK is a kinase enzyme which will add phosphate to the target proteins which are required in the particular cell cycle. They will act only when it is bound to cyclin molecules. CTK is consistently present in the cell, while cyclin molecules undergo a series of synthesis and degradation. We have a cyclin which is called as G1 cyclin. In vertebrates, it has a name called as cyclin D. In G1S, that is at the end of G1 and at the beginning of the S phase, we have a cyclin E. S phase, we have cyclin A, and in the M phase, we have cyclin B. This is in vertebrates. So, whenever the cell has to move from one phase to another phase, the particular cyclin has to be synthesized. It has to bound to your CTK, which will then get activated by various mechanisms, and then it will phosphorylate the target proteins which are required for the cell's progression in that particular cell cycle. So let's go to the options. Gradual increase in cyclin-dependent kinase activities in different phase of the cell. Gradual increase in cyclin-dependent kinase synthesis in different phase of the cell, or negative feedback loops to regulate the activity of cyclin-dependent kinases, or positive feedback loops to regulate the activity of cyclin-dependent kinases. First of all, I like to tell you that there is no feedback mechanisms in this particular process. The option A and B is left. What is the difference? In option A, they are asking there is an increase in cyclin-dependent kinase activity, while in option B, they are asking there is a increase in cyclin-dependent kinase synthesis. As we know, cyclin and CTK activities are low before the start of the particular cycle, and they go on increasing, and at the particular cell cycle it decreases and then it decreases. So there is a gradual increase in cyclin dependent kinase synthesis, or cyclin dependent kinase means your CTK. CTK molecule is consistently present, it is not synthesized. The molecule which is synthesized and it is regulatory component, that is cyclin. So your option will be first one, because there is a gradual increase in the cyclin dependent kinase activities. Let's move on to the next question. Question number 35. CTK1 can only be fully active when option A, it is phosphorylated on threonine 14. Option B, it is phosphorylated on tyrosine 15. Option C, it is bound to cyclin A. Option D, it is phosphorylated by CDC 25. This particular question is based on cell cycle regulation. If you want to learn in detail about this, you can watch my video on cell cycle as well as on cell cycle regulation. I will leave the link in the description box. Let us first understand the CTK molecule 1, it binds to your cyclin A, is correct. But when the CTK and cyclin binds to each other, their molecule doesn't get active, because there is one more protein called as a V1 which will add a phosphate at the tyrosine 15 or threonine 14, and this phosphate is known as the inhibitory phosphate. It will inactivate your component. Then you have one more enzyme which is called as GAT or CTK activating kinase, which will add a phosphate to your CTK at the 161 position of threonine. This phosphate is an activating phosphate. but still your complex remains inactive because here the inactivating or the inhibitory phosphate is still present. We require one more molecule called as CDC25, which is actually a phosphatase enzyme, that is an enzyme which removes your phosphate groups, and this particular enzyme will remove the inhibitory phosphate, and now this molecule is going to be your active form of your CDK. Thus, we have an option D, it is dephosphorylated by CDC. Only then your CDK molecule will be fully active. Let's move on to the next question. Question number 26. Which of the following cells surface marker is used to identify the B cells from the blood samples? Option A CD3, option B CD4, option C CD25, option D CD19. For this, we should know about the surface cell markers of different cells, and here it is mentioned about your B cells. Let's first understand about your stem cells. You know the stem cells, it is a progenitor of all your blood cells, and it is going to have a marker called as CD34. Then with using that stem cell, you are going to form your granulocytes, your monocytes, your T lymphocyte, that is your T cells, B lymphocyte, that is the B cells, and your thrombocyte, that is your platelets. Then each of these molecules have certain surface markers. CD45 is a surface marker which is present in all the cells which is derived from your stem cells. Then you are having CD15 in your granulocyte, CD14 in your monocyte, CD3 in your T lymphocyte, CD19 in your B lymphocyte, CD61 in your thrombocytes. These are the surface markers which help us to identify and differentiate between a particular blood cell. T lymphocyte also has two types of lymphocytes called as the T helper lymphocytes and your cytotoxic T lymphocytes, that is your TH and your TC cells. TH cell also has one more marker called as CD4, while your TC cell has a marker called as CD8. You have one more type of T lymphocytes which is when activated T lymphocytes, it is going to get your marker called as CDC25. The question is only asked about B cells. For your further information and study, I have given this particular image. You can take a screenshot of this and study all this. So we have answered here to identify B cell, your CD19, which is your option D. Shall we move to the next question? Question number 28. Loss of which of the following classes of molecules on the surface of a tumor cell target would result in reduced susceptibility to killing by host immune responses? As we saw in the previous question that there are certain surface cell markers which is going to help in your identification. Here we have given options CD3 and CD4 which are present in your T cells. Question is about a tumor cell target. See, a tumor cell is a normal cell, only that it has lost the control of its cell cycle and hence it is going to divide repeatedly. So it is like any normal cell. The option left is MHC class 1 and MHC class 2. MHC is nothing but major histocompatibility complex, as we know it. There are two classes of MHC. One is MHC class 1, another is MHC class 2. Now where are these found? As you can see in this particular figure, this is an antigen presenting cell which is an dendritic cell, and it has a marker which is called as MHC2. Now this MHC2 will present an antigen which will be identified by your T cells, thereby activating it to produce certain cytokines. Then as we can see, any infected cell or any cell in your body has one more particular MHC which is called as MHC1, which is identified by your TC cells using the CD8 surface marker, and this will allow your TC cell to produce certain enzymes called as granzymes and perforins which will destroy your infected cell. So you can now identify which is the correct answer. The correct answer will be MHC class 1, because MHC class 2 is present in your antigen presenting cells while MHC class 1 is present in all the normal cells. Next question. Question number 27. Administration of DPT vaccine, diphtheria toxoid pertussis product and tetanus toxoid would stimulate which of the following types of immunity? This is a very simple direct question. If you know the different types of immunity, you can answer it directly. But let me just revise it for you. We know immunity are two types. One is your innate immunity and second is your acquired immunity. Innate immunity is what? It is not specific to us, and we have studied a little bit about innate immunity in the previous videos. In case if you haven't watched your previous videos, I will leave that link in the description box. Let's move to the next, which is acquired immunity. That is, immunity is not inherent in an individual, but it is acquired by certain processes. There are two types of acquired immunity. One is your active immunity and the another one is your passive immunity. Active immunity will develop in response to an infection or vaccination. As we know, when we get infected, a particular bacteria or virus enters our body, which will then be identified by our immune cells, and then they will produce certain factors against them, and that's we become immune to it. Similarly, vaccine also works. Active immunity has two types. One is a natural one is your artificial one. Natural artificial will be because of an infection, while if it is because of vaccination, it is called as artificial active acquired immunity. Now, acquired immunity other type is a passive form. In case of active immunity, we injected the antigen and then antibody was produced by our own immune cells. While in passive immunity, we directly inject the antibodies. Recently, we all have heard about this coronavirus pandemic that there is a plasma therapy. What we are doing in plasma therapy? We are taking the antibodies which are produced in response to the coronavirus by the infected person, and that particular antibody containing plasma is transfused to an individual who is currently suffering from the infection, and it is found that this particular antibodies was effective in controlling the infection in many individuals. So that is an example of passive immunity. So here we directly give the antibodies. We don't give any work to your immune system. So there are two types again. We have natural as well as we have artificial. In the natural, we get it from your mother. Like when we see infants, when a mother feeds the infants with the breast milk, that breast milk contains antibodies, and that antibodies will directly be helpful to infant. While in artificial, we get certain immunoglobulins, or the currently as we know the plasma therapy, where antibodies are directly infused. We have understood the basics of immunity. Let's come back to the question. We have asked a question about DPT vaccine. We know DPT vaccine is given to every infant once is born. This particular vaccine can prevent the infection against three particular diseases. One is your diphtheria, one is pertussis, that is whooping cough, and other is tetanus. So we have a toxoid vaccine because in diphtheria as well as in tetanus, a particular toxin is released by the infecting organism, and that we have a toxoid, which is an inactivated form of the toxin. Secondly, we have certain products of this organism which is causing the whooping cough. So we will inject this into the individual. That individual immune system gets activated, and that then ensures that the particular immune system will produce antibodies against these three different types of infectious agents, and the particular person who received that particular vaccine is immunized. Now, can you identify which of this is it? As we know, since we are injecting it and we are giving antigen but not an antibody, so it will be active. So now there are two options: either it is artificial active or it is natural active. We know artificial active is when we have a vaccination, and natural active is when we have a disease. So the answer will be artificial active. Very simple. Let's move on to the next question. Question number 21. Genes for 16S and 28S rRNA are transcribed by dash. Okay, this is again a very simple direct question. First, let us understand what is the 16S and what are the 28S rRNAs. First of all, rRNAs are ribosomal RNAs, that is RNAs which are present in your ribosomes. As we know, we have ribosomes in prokaryotes as well as in eukaryotes. The ribosomes of prokaryotes are 70S, while the ribosomes of eukaryotes are 80S. First, let's discuss about the ribosomes of your prokaryotes. That is 70S consists of two subunits. The largest subunit, it is a 50S, which consists of the 5S RNA and 23S RNA, while 30S subunit contains your 16S RNA. So your question is about your 16S RNA. Let's come to eukaryotic ribosome, which is having two subunits. The largest subunit is 60S and the smaller subunit is 40S. Your 60S subunit contains your 5S and 28 RNA along with the 5.8S small RNA. In the small subunit, we are having the 18S. So here the question is asked about your 28S RNA. In prokaryotes, we have only one type of RNA polymerase. Whenever there is transcription, we know transcription is carried out by RNA polymerase. Now, many students have the confusion if it is DNA polymerase or RNA polymerase. They can confuse between replication and transcription. I just give you one particular example for this. See, we know during transcription what happens, we are converting our DNA or we are transcribing our DNA and producing RNA out of it, right? So, the polymerase enzyme is named like this. It is called as a DNA dependent RNA polymerase. It's called as what? DNA dependent RNA polymerase. So, in transcription, the polymerase will be RNA polymerase. While during a replication, we know that DNA is replicated to form another copy of DNA itself. So here the polymerase will be called as DNA dependent DNA polymerase. So here the polymerase will be DNA polymerase. So we now understood that our answer will be an RNA polymerase. As we know, we have three types of RNA polymerases, RNA polymerase 1, 2 and 3. And now let us understand what all these particular three RNA polymerases encode or transcribe. Prokaryotes, only one type of RNA polymerase which consists of 2 alpha subunits, 1 beta and 1 beta subunits, and this requires a sigma protein for initiating as a promoter. It also requires a rho protein to terminate, and then prokaryotic RNA polymerase is inhibited by rifampicin and actinomycin T. Now in eukaryotes you have RNA polymerases 1, RNA polymerases 2 and RNA polymerase 3. RNA polymerase 1 is going to transcribe your rRNA except your 5S. So there is a 5S RNA which is present in both of them. It is not transcribed by your rRNA. While RNA polymerase will transcribe your HN and your mRNAs and some of the snRNAs, while RNA polymerase 3 is going to transcribe your tRNAs and the 5S rRNA. Let's now understand, in eukaryotic RNA you don't have any sigma factors, but there are certain transcription factors which bind to RNA polymerase and initiates its transcription, while there is no rho molecules for the termination of transcription. Similarly, here RNA polymerase 2, which is very important for the transcription of mRNA and it is inhibited by alpha amanitin, which is present in your mushrooms, and actinomycin T. There are various questions asked about your amanitin and actinomycin T which will inhibit your RNA polymerase 2. So we have our answer, that is rRNA synthesized or transcribed by RNA polymerase 1. Let's move to the next question. Question number 31. Which type of inhibition requires binding of one or more substrates to enzyme before the inhibitor can bind? We know enzyme and substrate bind together to form enzyme substrate complex, and later on it splits to form your enzyme, as well as enzyme is released and your product is formed. This is a normal type of an enzyme substrate reaction. Let us now understand about the inhibitors. There are two types of inhibitors. One is your reversible inhibitor and second is your irreversible inhibitor. Reversible inhibitor, whenever it binds to your enzyme or your enzyme substrate complex, it will inhibit the enzyme. But this inhibition is transient, that is, your enzyme can again become active. While irreversible inhibitor will completely destroy your enzyme, it can never become active again. Now this question is actually about your reversible inhibition. You have around 4 types of inhibition. One is uncompetitive, non-competitive, mixed inhibition and competitive inhibition. Now let's understand briefly about all this before going on to solving the question. Now as you can see here, this is the first one is your competitive inhibition, wherein this I, which is called as an inhibitor, is binding to your enzyme and it produces the enzyme inhibitor complex. Now this enzyme inhibitor complex cannot form the product. Okay, so it is going to block your enzyme. Now in case of competitive inhibition, your substrate as well as inhibitor have certain sequence similarity, hence your inhibitor can bind to the active site where actually substrate has to bind, and it will prevent or it will compete with your substrate to bind to the active site. So hence this is called as competitive inhibition, where the inhibitor binds to the enzyme's active site. Secondly, we have uncompetitive inhibition, where your enzyme is there, it binds to your substrate and form ES complex, but here this particular inhibitor cannot bind to the enzyme directly. It will only bind after this enzyme substrate complex is formed. Since here it binds to inhibitor, this particular complex becomes inactive. This happens in case of uncompetitive inhibition. Let us understand non-competitive inhibition. In non-competitive inhibition, your inhibitor will bind to your enzyme. Your inhibitor can also bind to your ES complex. So it is twofold. So your inhibitor will bind to enzyme also. It will also bind to any ES complex it would have formed in the reaction, thus inactivating your reaction, and there will be no formation of product. In the mixed inhibition, again this is similar to your non-competitive inhibition. Your enzyme binds to the inhibitor. Your ES complex also it can bind, and even this ES complex can also forms product. Okay, now let's come back to the question. The question is, which type of this inhibition requires binding of one or more substrates to the enzymes before the inhibitor can bind? Definitely it will be your uncompetitive, because both in uncompetitive and your non-competitive, inhibitor binds to your enzyme substrate complex. In case of non-competitive, the inhibitor can also bind to a free enzyme. Hence the answer will be uncompetitive inhibition. Let's move on to the next question. The genes which remain confined to differential region of Y chromosomes are dash. So this particular question is about Y chromosome. So let us discuss about your X chromosome and Y chromosome in detail. As we know about human beings that we have total 46 chromosomes, that is 22 pairs of autosomes and pairs of sex chromosomes. If you are a female, you will have pair of X chromosome, and if you are a male, then you will have a one X chromosome and one Y chromosome. Now the question is, why only males have Y chromosome? Because Y chromosome can only be passed from the father. Let's compare both of this X and Y chromosome. We just can, by just looking at it, we can say that Y chromosome is very short as compared to the X chromosome. Secondly, if you can see the arrangement of centromere over here and here it is over here. Okay, so that's again a difference. But during meiosis, we know there is a crossing over between all the chromosomes, and crossing over also takes place between X and Y chromosomes. But as we know that crossing over can only take place between homologous chromosomes. So since these two chromosomes are completely different, but there are certain regions that's called, this marked this red color, you can see in both the cases, which is called as your pseudo-autosomal region or your PAR region. This is a primary and this is a secondary PAR regions. Pseudo-autosomal regions, they have a homologous sequences where actually crossing over takes place. Now all the other regions which is marked in this Y chromosome green color are the chromosomes which are unique to your Y chromosomes. They are not present in your X chromosomes, and that is a question all about the differential region of Y chromosome. It means the region of Y chromosome which is not present in your X chromosome, it is called as holandric genes. So those genes are called as holandric genes. We also one particular gene which is present in this holandric gene called as SRY gene, which is very important, which will determine for determining of male-like character. The main important gene is SRY. Okay, this you might have in some other question, so I'll just mention it here. So we have got the answer to this question. Let's move on to the next question. Question number 33. Blastopore is dash. Option A, opening of neural tube. Option B, opening of gastrocele. Option C, future anterior end of embryo. Or option D, found in blastula. This question is about developmental biology. We have a zygote which is formed by the fertilization process of the ovum and the sperm, and once fertilization is complete, zygote is formed. Zygote will undergo cell division. There is a different set of cell division, 2 cell, 4 cell, then there comes to your 8 cell. We have all those morula stages, and we can see there is a 8 cell stage, and once after this particular cells, 8 cells will undergo more cleavage, that is it will undergo more cell division to form something called as blastula, and this blastula looks like a empty ball. It is like a tennis ball which is empty inside, and outside it's having a layer of cells. And then after this process of formation of blastula, there is a formation of gastrula, and gastrula formation starts with a process called as gastrulation. And what happens is particular cells will move inside, and then they will form a layer over here, and that is forms like this, and then these particular layers will get differentiated to form your ectoderms, your endoderms and archenteron. Archenteron is actually just nothing but your gastrocele, and the outer man we can see here, this is your blastocele. Okay, now let's come to the question, what is blastopore? Where you can find blastopore over here. So blastopore is actually an opening like thing in some particular organisms which are called as protostome, where this particular blastopore will form the mouth, wherein some deuterostome organism this will form the anus. Now this particular is opening of what? It is the opening of something called as archenteron. Okay, as you can see, it is the opening of this archenteron. Now this archenteron is, other name of archenteron is nothing but gastrocele. So the answer is, it is an opening of gastrocele. And now I would like to say thanks for watching this video. In the next video, I will be solving many more questions of this GAT-B entrance examination. I will also make a further video regarding your section A questions. So like this video and share it to your friends who are preparing for this exam.